So let's have a look at Plastid transformation. So your basic requirements is a method of delivery, and by and large people have used biolistics. It is possible to use protoplasts, but the bialistic method works well and is easier to work with than protoplasts. And since the initial successful transformation in 1988 with Chlamydomonas, it's been repeated in a large number of other species, but it's still only routine for tobacco. Now, biolistics will get your gene into the cell and into the chloroplast, so this is great. So virtually all your protocols are going to use biolistics. Some will use protoplasts, but most will use biolistics. Once your gene is in, then you need a good selectable marker in order to be able to select for your particular gene. The initial studies used the RRN16 gene, which is a 16S ribosomal RNA gene in the chloroplast. And you can get, this gene is susceptible to spectinomycin. So if you add spectinomycin to wild type cells, they will actually die off because it inhibits this 16S ribosomal RNA. However, a point mutation will actually confer resistance. So it's possible just to put in a single piece of DNA with this point mutation in it. It will go in by homologous recombination, and you end up with a spectinomycin resistant variant. So the initial studies on Plastid transformation use this RRN16 gene. Now, it's not a very effective way of actually producing resistance. The first reason for this is because it only actually acts on the actual site of action. So it's not doing anything about detoxifying the spectinomycin. It's just acting at the site of action on the 16S ribosomal RNA subunit. The other problem is you get lots of escapes. So the mutation rate and the translocation rate in chloroplasts is absolutely phenomenal. And what happens is that you end up with either the 16S ribosomal RNA point mutation actually reverting back, or much more commonly you end up with false positives because of random mutations in the ribosomal RNA RRN16 gene. Far better is to actually put in some kind of detoxification method for spectinomycin. And this detoxification method is through this AADA gene, which is the 3-adenyl transferase gene. So you'll see that most protocols for Plastid transformation nowadays will put in the AADA gene as a selectable marker. So this will actually adenylate spectinomycin and therefore make the spectinomycin inactive. And you can see here the construct of the gene. So the construct has got the -35 to -10 region, which is the promoter region, and also a region where you find the Shine-Delgado like ribosomal RNA binding site. It'll also have 5' untranslated region. These are regulatory elements, and also a 3' untranslated region. These are regulatory elements that can actually help the expression of the ribosomal RNA. So you can get 100,000-fold variation in expression depending on these 5' and 3' untranslated regions. So you'll see that when they're developing and designing their plasmid, not only do you have to worry about which particular selectable gene you're going to put in, but you also need to worry about these untranslated regions, the 5' and 3' untranslated regions. So they tend to go for things like the PSBA gene, 5' untranslated region, the TPSBA 3' untranslated region. And you can see they've got in there the AADA open reading frame and a Shine-Delgado sequence for ribosome binding. And you'll also need some sequences for homologous recombination. So outside of this particular vector that you're designing, you need to select a left and right hand side targeting region to allow the gene to go in by homologous recombination. Okay, and this neatly describes how the gene goes in. So you've got two genes and an intergenic region there, and the flanking regions are homologous to this intergenic region. And you've put in there two genes, the AADA gene is a selectable marker, the Cry2A AA2 gene, which is for Bacillus thuringiensis crystal toxin. Now, when you're looking at your promoter, it's important to have a very strongly expressed promoter in order to get expression. And some of these promoters are very highly expressed in green tissues, but they may not be expressed well in non-green tissues. So you have to look at where do you want this gene to actually be expressed. So there's a question mark there, not expressed in fruits. So some of these genes may not be expressed in, for example, etioplasts, which are not green, or in amyloplasts, which are not green. This is just a table to have a look at expression levels. So you can see this is the crystal toxin single gene expression in A for nuclear transformation, and you can see that around about 0.35% of total soluble leaf protein is this Cry2A A2 Bacillus thuringiensis crystal toxin. If you compare that to expression in plastids, we've specifically targeted it to chloroplasts, you can see that in leaves you're getting almost 50% of total soluble leaf protein expressed as Cry2 Bacillus thuringiensis crystal toxin. So it is possible to produce phenomenally high levels of expression in transgenic, transplastomic plants. One of the things that you have to do when you're selecting, you need a very rigorous selection for your transplastomic plants. The reason for this is that any one cell will contain numerous copies, numerous chloroplasts, so you may have 60 or 100 chloroplasts per cell. Each chloroplast will contain multiple copies of plastid DNA. So you can see here at the top left, you've bombarded your cell and you've got the plastid DNA into the cell. But the cell contains more than 10,000 plastid genomes per cell, because of the large numbers of chloroplasts, the large numbers of plastid DNA per chloroplast. So you get this into a single chloroplast. You then need to start selecting. So initially you'll start to get overexpression of your gene when you're selecting for it. And you can see here we've actually reduced the number of chloroplasts, so we're going through proplastids instead. So instead of one chloroplast that's got some transgenic DNA in it, in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 chloroplasts, we've reduced the number of chloroplasts simply by going through proplastids rather than chloroplasts. So we're selecting one chloroplast out of 4 chloroplasts rather than 1 out of 10. And then you carry on with your selection and you finally end up with the gene stabilized. After several cycles of antibiotic selection, you end up with homoplasmy rather than heteroplasmy that we have here. And this is an example of selection of transplastomic clones by spectinomycin resistance. So we've bombarded a whole load of leaf strips and we're getting the gene expressed. You can see here all of these leaf strips have been, they're yellowed because of spectinomycin affecting the 16S ribosomal RNA, whereas one small cluster of cells has actually escaped the spectinomycin because it's been genetically transformed and it's regenerating into a plantlet. And then when we plate these out after a number of generations of selection, this is the transgenic tissue, this is wild type tissue. So to finish up, if we do a comparison of the nuclear and plastid genomes of angiosperms, a nuclear genome, 2 copies of each, many chromosomes, you could end up with thousands of genes per chromosome. But importantly, the arrangement of transcription of genes, each gene separate, individually transcribed, whereas you can have operons with plastid DNA.